So we'll go ahead and get started. Thanks everyone for coming. It's really my honor and pleasure to have Dr. Christopher Newton-Cheh here. Chris really is a world leader in the genetic epidemiology of cardiovascular disease and lives really at the interface between basic science discovery and clinical translation, sort of embodying expertise in epidemiology, genomics, molecular genetics, and clinical cardiology. So a very unique individual who has been extremely successful early in his career. Chris went to med school at Columbia, where he graduated AOA, did his residency and was a chief resident at the MGH, did his clinical cardiology fellowship at the MGH, did a master's in public health at the Harvard School of Public Health, and then did two postdoctoral fellowships, one with Chris O'Donnell in epidemiology at the Framingham Heart Study, and then one with Joel Hirschhorn at the Broad Institute. So really cool kind of mixing of basic science and clinical translation. Harvard was lucky enough to retain him on faculty, where he is now a clinical cardiologist at the MGH, specializing in heart failure transplant. He's medical director of the ECG lab. He's a faculty member at the Center for Human Genetic Research at MGH and is a scientist at the Broad Institute. He's on the scientific advisory board of the FDA Endocrinologic and Metabolic Committee. He's a member of the Cardiac Safety Research Consortium of the FDA Pharma Academic Roundtable, and he's chair of the program committee of the AHA FGTB Council, so very accomplished national leadership. His research laboratory focuses on the genetics of hypertension, sudden cardiac death, and cardiotoxic drug responses, and he has over 130 publications, including Nature Genetics, New England Journal, JAMA, and he's had a couple of papers on genetics of blood pressure that have been cited, one as the top cardiovascular discovery of 2009 by the AHA, and one as the top original paper of 2013 by Hypertension. So very accomplished investigator, and we're lucky enough to have him today talking about harnessing genetics to identify druggable targets and causes of off-target cardiotoxicity. So thanks, Chris.
Thanks very much, Swati. That's a great introduction. I wish I could record it and play it so that my wife and my kids could appreciate that someone thinks I could do something that counts. I don't have any specific conflicts of interest related to what I will discuss today, although I do have a patent related to a microRNA, but I will not be talking about any clinical implication of the work, nor is it approved. As Swati mentioned, my laboratory uses population genetic studies of blood pressure and arrhythmia risk factors, specifically QT interval variation. And we, in addition to the population genetic studies, use genotype-directed studies in people and through collaboration with molecular biologists attempt to identify underlying molecular mechanisms. There'll be two parts to my talk. The first part will be what I'm calling identification of targets for antihypertensive therapy, basically a summary of blood pressure genetics and kind of the highlights. And the second part will be related to identification of markers of off-target cardiotoxicity, and specifically discussing the QT interval genetics. As this audience will well appreciate, blood pressure is a major cause of stroke, myocardial infarction, heart failure, and kidney disease. Control of elevated blood pressure with antihypertensive therapy lowers the risk of stroke, myocardial infarction, and heart failure, and the kidney disease story is probably the weakest among them. But blood pressure is thus a causal risk factor for those traits, and therefore I think reasonable to be interested in its genetic determinants. Blood pressure, in fact, has a continuous and graded influence on risk without a clear threshold in effect, even down to as low as a systolic blood pressure of 115. Each 10 millimeter of mercury increase in blood pressure is associated with an increase in cardiovascular disease risk. Guyton's model of blood pressure regulation may be known to many of you from med school. This is not designed for you to be able to recognize the different modular components in this schema of the factors that contribute to blood pressure regulation. This is a snapshot from about 20 years ago, and it may seem overly complicated. I'll come back to that. Human genetics can identify novel mechanisms regulating blood pressure, and specifically Mendelian hypertension and hypotension syndromes have been exploited by people like Rick Lifton and others to highlight the role of rare mutations in renal salt handling genes in regulating blood pressure. This has been critical because many of the components of renal salt handling have been intervened upon with antihypertensive therapies, such as those that target the ACE enzyme, the angiotensin II receptor, aldosterone, and the targets of diuretics. So clearly, there's a correspondence and a translation of human genetics of blood pressure syndromes to therapeutics. But what about common variants? In this talk, I will describe several aspects of some studies of common variants that I think illustrate what blood pressure genetics can tell us further beyond continuing to solve the Mendelian syndromes, which within the next 5 to 10 years likely will all have been solved. So first I'll talk about a genetic variant that influences natriuretic peptides and blood pressure, and a follow-on genotype-directed physiologic study. Then I will summarize all of the GWASs of blood pressure to focus in on one pathway, blood pressure regulation in humans. And this is work that I led with Tommy Wang, who's now the chief of cardiology at Vanderbilt, but was at Mass General at the time. Tommy and I, in fact, were fellows together. The conclusion of that work was that we found a common polymorphism, RS5068, and I will try not to use too many SNP IDs, that was associated with ANP levels as well as blood pressure in humans. This minor G allele of that SNP. It's an AG SNP, so everybody has 2 copies of the ANP gene that you've inherited from your parents. Most of you will have 2 copies of the A allele. About 7 or 8% will have an A and a G, and then about 1% or fewer, if you're of European ancestry, will have 2 copies of the minor allele. And if you're South Asian or of African ancestry, it'll be closer to 1 in 1,000. Increasing copy of the minor G allele is associated with higher A and P level, lower systolic blood pressure, lower diastolic blood pressure, and reduced odds of hypertension. And just to orient you to the p-values, I would say for this talk, if I present an association, I will report it as significant if it has a p-value less than 5 times 10 to the minus 8. So if you're doing a clinical trial where you randomize people and you have a trial designed to test a specific hypothesis, a p of less than 0.05 means you have a chance of 1 in 20 that you'll observe that result due to chance. In the case of a genome-wide association study, there are effectively about 1 million independent common variant tests in the genome. So if you use that 0.05 threshold, you divide it by 1 million, you get 5 times 10 to the minus 8. I'll make this point a couple times because I think clinical people can get confused. Higher ANP or higher BNP is bad if you're in the emergency department with shortness of breath, but it's potentially good given the biologic properties of ANP, and in this case, it's genetically determined. So we sought to identify individuals based on their genotype for this SNP and to perform higher resolution physiologic profiling in individuals with the high versus low ANP genotype. And I'll say that in this study here where we did the ANP study, the reason we got to this p-value of 10 to the minus ridiculous was that we had 15,000 people in whom we had measured ANP. It took 30,000 people to achieve these, by comparison, more modest p-values, although this has subsequently been replicated. This is, and I'm not going to go into it in detail, this is blood pressure that's observational in people on random salt diets who are taking all sorts of medicines, potentially smoking up until they come into their epidemiologic cohort. So it's a pretty messy phenotype. Our hope was by doing a much cleaner analysis of a smaller subset of people, we'd be able to discern underlying effects. And this is work that was led by a talented postdoc in the lab, Pankaj Arora. He's now at the University of Alabama as a cardiology fellow. Tommy and I both did the clinical work. Ken Block led the lab in which Pankaj did the molecular experiments. Ken actually passed away earlier this month from cancer, and I think we're all missing him dearly. For the design of the study, we genotyped 700 people of European ancestry. We actually have recruited about 1,400 people in a multi-ethnic recruitment of healthy volunteers. These are healthy people who had normal blood pressures, normal body mass index, with ages between 18 and 40. So we specifically chose people who were less likely to have already developed hypertension or already be on medications that might interfere with our study. We identified people based on their genotype. We got 23 people with the more common AA genotype through the study, and 8 people with the AG genotype. That was the one associated with the higher ANP. And we put all of those people, they were in random order, put on a high sodium diet or a low sodium diet. 4.6 grams a day is a typical McDonald's American diet. 0.23 grams a day for anybody who takes care of heart failure patients. This would be your fantasy to get patients on this low a sodium diet. It's a 20-fold difference in sodium exposure. And then at the completion of each of those weeks of the diet, they had an overnight stay. We got recumbent blood pressures checked overnight. We did some recumbent aldosterone renin testing, and then gave them a couple liters of intravenous saline in a weight-based manner, but it worked out to about 2 liters over 2 hours. That's about 300 milliequivalents of sodium. 0.23 grams a day is 10 milliequivalents of sodium, so it was a big stimulus. And we measured their NT-proANP, that's the N-terminal propeptide of ANP. I won't belabor that. So it was a complicated study. We had to, people had to be able to take off work. They had to eat the food that we provided. I would say 31 seems kind of paltry, but it took a bunch of work to get those people in. And by design, those people were young, median age of 25, had a they were a normal BMI, normotensive. It was mostly men. We had to time it with the menstrual cycle, and that added an additional logistical challenge. 8 out of 10 of the people who completed only one of the diets were women because we just couldn't get them back to juggle it with their work schedule. All right. So this is a summary of the effect of diet on and of genotype on plasma ANP. And this is way below where we're detecting ANP in the clinic in patients with heart failure. There's about a 50% higher ANP in the 8 people with the AG genotype compared to the 23 people with the AA genotype. Similar effect on either of the 2 diets. On the low salt diet, the ANP is lower. Salt is either chronically or acutely is a stimulus for ANP production and release. And so the effect of the genotype on ANP is about comparable to the effect of a 20-fold difference in dietary sodium, pretty dramatic sodium restriction. And we did confirm that all these people were in steady state for sodium. This is the results during the saline challenge. We found while the baseline differed, whether you were on a low salt, higher on a high salt, for the AG, that's the open triangles on top, and the solid square AA on the bottom, you'll see that both groups had a rise in their ANP over it was a 2-hour infusion and 6 hours of continued sampling. But in fact, the genotype did not alter the response to intravenous saline. In fact, neither did the diet. So there may be a suggestion of a slightly steeper curve here, but being on a high salt or a low salt diet did not appear to interact. So essentially, there's a genotype effect, there's a dietary sodium effect, and there's an acute saline response, but all of those were independent to each other. BNP differs on a low versus a high salt diet. BNP rose over the course of a saline infusion. This is NT-proBNP, but it did not differ by genotype, and we already knew that from our 15,000-person study. So in a genotype-directed physiologic study, standardization of the salt background refined the association between genotype and plasma A and P. The effect of genotype was pretty similar to a marked change in salt intake, but the genotype did not alter the A and P response to dietary or intravenous sodium. In fact, this was my hope, that we would find an interaction between the genetic effect and the dietary exposure, and we did not. In data that I will not show you, we did also report in this paper that we identified a microRNA, microRNA 425, as a negative regulator of A and P expression, and we demonstrated that, in fact, the SNP that we were focused on in this study was a causal variant and identified a mechanism of action. All right, so now comes the summary of many years of blood pressure genome-wide association studies. There were a couple that were published in 2009. These were the first to identify reproducible associations. From 1990 to 2008. There were irreproducible candidate gene studies that littered the field. These were studies that started with 30,000 people with the genome-wide association data. Our study had 70,000 people for replication. I'm not telling you so you'll be impressed by the numbers, but I'm telling you so you'll be daunted by the numbers required to achieve significance. And I'll explain more about that later. We followed up by joining forces between those two groups, and there's a general theme of the field of collaborate, build, and continue to collaborate with anybody you can find. And this is the latest summary we have from a paper that was in Nature a few years back. This was 365 co-authors, and by that it's just to describe, I guess from a DCRI perspective, it's probably not a lot of people, but it took hundreds of institutions to accomplish this research project. The starting genome-wide association study was about 70,000 people. We did replication in 130,000 people of targeted SNPs. So the total sample size was up to 200,000 people. This study added to prior findings and brought to the total about 28 genome-wide significant polymorphisms. If you take all 28 of those polymorphisms and you take each one has an effect of about a 0.5 millimeter of mercury systolic blood pressure, a 1/4 millimeter of mercury diastolic blood pressure. That would be a failing drug if that was the difference you got in a large trial. These are individual genotypes. This is why it took us scores of thousands of people to find these. If you bundle their little 0.5 millimeter this way for SNP A and 0.5 millimeter this way for SNP B together into a score, then you can predict what the change in blood pressure is on the basis of those 28 polymorphisms for any given individual. It turns out that 28 SNP score does not explain a lot of blood pressure variability. It explains about 1% of variation in blood pressure, which is a very heterogeneous trait, and I'll come back to that. If you take the top 20th percentile of that score compared to the bottom 20th percentile, well, you shouldn't be surprised that it's associated with a higher odds of hypertension. These are SNPs that influence blood pressure. That's kind of to be expected. But they also have an increment in risk of coronary artery disease, of stroke, and of left ventricular mass, which is a risk factor for heart failure. These, I think, are all as expected from decades of study of antihypertensive therapy and of Mendelian hypertension families. There was no effect on estimated glomerular filtration rate. We didn't have a large enough sample with end-stage kidney disease. I did mention that clinical trials are less overwhelming about the benefit of blood pressure lowering therapy. For end-stage renal disease, yes, but for chronic kidney disease, it's less clear. So this is a summary across the genome of the results. We put little flags here for genes nearby, the SNPs that we found. This is not an informative slide other than to be a way for me to represent a few of the findings that I'll highlight. One of them is a SNP at a locus that includes NPR3 that in fact encodes the natriuretic peptide receptor C. This does not have guanylate cyclase activity like the ANP receptor, which is natriuretic peptide receptor A, but the knockout results in lower blood pressure and skeletal overgrowth in a mouse model. I guess I do have a few mice strewn in here. A highly correlated variant is associated with height from independent work, such that the higher BP allele was associated with shorter stature, consistent with the knockout directionality. But when we looked in humans, we couldn't find association of the variant with plasma ANP or BNP or NT-proBNP in about 7,000 people. Makes it unlikely that it's acting through blood levels of natriuretic peptides. It could be some tissue-specific local effect. In fact, neprilysin is responsible for the degradation of active BNP and ANP. It's possible that that compensates for it. So we don't have yet an explanation. This is the results genome-wide for diastolic blood pressure. I'll just highlight one labeled GUCY here. It's labeled GUCY because it's a SNP that sits in a region with two subunits of the alpha-1 and beta-1 subunits of soluble guanylate cyclase. It is a weak genetic effect, but if you have enough people, 200,000 people, you can get to a p-value that's recognizable as not spurious. Soluble guanylate cyclase is the protein that transduces the effect of nitric oxide. Nitric oxide is produced by endothelia, diffuses across cell membranes, and in smooth muscle cells leads to vascular relaxation. Mouse knockout of both the alpha-1 and beta-1 subunits leads to hypertension, so it was not surprising to find a result in this region. In fact, soluble guanylate cyclase activity is reduced in pulmonary hypertension and targeted for pharmacotherapeutics, and now on the market is soluble guanylate cyclase activator, riosuguat, for the treatment of pulmonary hypertension, either idiopathic pulmonary hypertension or that due to chronic venous thromboembolism. Another SNP is it's in fact, all the SNPs I've described so far are non-coding. They don't encode an amino acid change, but it lies in the region that includes ENOTH, the endothelial nitric oxide synthase. This is a study using a subset of the genome, just a tenth of the genome was captured on this genotyping array, but they found a common variant associated with a weak effect on diastolic blood pressure. Again, mouse knockout, hypertension, so not a surprising gene to find. And lastly, in unpublished work, we've done analysis of 200,000 SNPs on a metabochip array in 200,000 people. We're trying to finish up the paper. It takes a long time to write these papers with so many components. We found it's now 22 novel loci because in the time when we originally had the discovery, there have been other publications. One of the loci includes phosphodiesterase 3A. In fact, that is involved in the metabolism of both cyclic GMP and cyclic AMP. We found in this study independent signals at the natriuretic peptide locus, the guanylate cyclase locus, and PDE3A. And in total, we have 66 common variant blood pressure loci that have been discovered, either published or unpublished. We've also done a GWAS of natriuretic peptides and found additional loci, a couple of which are already established blood pressure loci. So we've already found the likely mechanism underlying a couple of the blood pressure loci I've described. We're in the process of writing that up. So hopefully I've showed you that these cyclic GMP pathways, both soluble guanylate cyclase and natriuretic peptides, converge on the production of cyclic GMP. A potential implication of this is that one could consider antihypertensive therapies that target the production of cyclic GMP, whether through natriuretic peptide receptors or through the soluble guanylate cyclase. There are active programs to develop oral natriuretic peptide agents that are intended to treat hypertension, and it's a consideration, but probably since there are many other good antihypertensives, most of the pharmaceutical companies that are looking at SGC stimulators and activators are targeting pulmonary hypertension, at least to get on the market. One could consider targeting therapies on the basis of genotype. It's possible that people who have a genetically determined low SGC state could be targeted for SGC therapies, but frankly, I think that's unlikely. I think it may actually be the case that the drugs that target SGC, for example, they may have a differential effect based on genotype, but I'm dubious that we'll be selecting a segment of the population who should get those therapies on the basis of this genotype. The blood pressure effect is sufficiently modest. It's hard for me to imagine that these people have a uniquely SGC deficient cause of their hypertension. Among all 66 common variant loci identified to date, how many are in known antihypertensive targets? It's a reasonable question if we're talking about using genetics for identifying drug targets. Here are all the currently marketed agents and their classes, and there are thiazide diuretics, renin-angiotensin-aldosterone inhibitors, beta blockers, calcium channel blockers, and direct vasodilators. Among the 66 loci to date, there's one that includes a calcium channel subunit, one that includes angiotensinogen, which I count here as being the ACE inhibitor ARB pathway, and one that's the beta adrenergic receptor, a target of beta blockers. How many of the loci, the 66 loci, include Mendelian genes? One. Cytochrome P450 17A1, encoding the 17-alpha hydroxylase, is associated with Mendelian hypertension. So what does this tell us? Why so few blood pressure SNPs and established drug targets are Mendelian genes? I think the reason is that Guyton's wiring diagram is in fact oversimplistic. This is a much more complicated system than this simple diagram would suggest. And in fact, I think this is the big take-home for me of the blood pressure work. There are probably thousands of common variants of equally or weaker effects on blood pressure. And I think our understanding of blood pressure regulation is incomplete. The fact that these 66 little darts in the genome only lined up occasionally with things that we know about blood pressure tells us there's a lot we don't know about blood pressure. It also tells me that we're unlikely to be using genetics, at least in the foreseeable future, maybe 10 years from now, 15 years from now, it'll be different, to get a fingerprint of your specific type of hypertension. I just don't see it happening given these weak effects. But these are novel therapeutic opportunities. If you can identify the mechanisms, then, or even repurpose existing therapies, have a drug that's targeting the treatment of pulmonary arterial hypertension, maybe you can also target systemic hypertension unless the toxicity profile is off. 7 out of the 66 loci are involved in cyclic GMP signaling. This, to me, is the real standout. I think there are lots of lists of candidate genes, natriuretic peptides, NO, SGC were on those lists in the 90s and in the 80s. But this really stands out. This seems like a dramatic overrepresentation of one converging set of pathways on cyclic GMP. I think that's really the take-home lesson from the blood pressure work so far, that the systems involved in regulating intracellular cyclic GMP are important for blood pressure regulation in humans. The challenges here are that we need to find the targets, we need to identify causal mechanisms, we can try to identify coding variants that have strong effects using rare variant approaches. We need to determine how specific these genetic effects are. We can consider human studies and animal or cellular models where they recapitulate the human, but it's challenging. And genotype-directed physiologic studies, the only reason we were able to be partially successful with the natriuretic peptide study is we could measure the blood biomarker that is the intermediate for the blood pressure effect. If we went after blood pressure with our sodium intervention, we would have need 500 to 1,000 people. Targeted sequencing, we've already done this. We did find another gene. Whole exomes, we haven't published it yet, but we're writing it up. Whole exome sequencing is another potential approach. Low-frequency variant arrays, in fact, a low-frequency variant array exome chip would have detected our sequencing discovered finding. Looking at variants that we found in relation to blood pressure for relation to pulmonary hypertension or preeclampsia is yet another approach. These are other hypertensive syndromes. All right. So part 2, and in 15 minutes, I'll try to summarize the QT interval work. So sudden cardiac death claims about 300,000 lives annually in the United States. Some of it is due to ventricular fibrillation and ventricular tachycardia. Coronary disease is common. It's present in about 85 to 90% of people who die suddenly. But risk factors have been inadequate to predict who will die suddenly. And sudden death is the first clinical event in up to a third of people who die suddenly. One of the components to sudden death is QT prolongation, which at the population level is a risk factor for sudden death. Probably some or a substantial portion is mediated through torsade de pointe, which is a form of ventricular tachycardia associated with QT interval prolongation. This arrhythmia is a hallmark of drug-induced long QT syndrome. There are about 80 currently marketed therapies. 15 have been pulled from the market because of this toxicity. As little as 8 to 10 milliseconds of QT prolongation has been associated with enough arrhythmia at the population level that the risks and benefits have not favored continued marketing. But for example, some oncologic therapies such as arsenic, prolonged Dan Roden had shown that the frequency of this polymorphism was 7% in those who had drug-induced arrhythmia compared to 1%. This is actually in an abstract and a patent application. The paper was never published from this work. A small study of drug-induced arrhythmia found a slight excess, but these are pretty borderline on their own. Studies of those who were mutation negative for known LQT1, 2, and 3 genes had overrepresentation, 9 and 11% frequency for this polymorphism compared to about 2.5% in controls. And in a Japanese study, it was also shown to be overrepresented, 7.5% compared to 0.8%. So this polymorphism associated with about a 10 millisecond difference in QT interval, again, that was about the threshold where the FDA spidey sense goes up about whether the drug should come on the market or be subject to increased regulatory scrutiny. And that translates into an impact on drug-induced arrhythmia and congenital long QT syndrome. Stefan Kabe's group published a study of 176 patients with torsade. Many of those had been exposed to QT prolonging drugs, but this is ascertained on torsade. They studied 18 candidate genes. One of the SNPs included was this D85N polymorphism. It had a high odds ratio in their initial study, a high frequency compared in mutation. They got 205 mutation carriers. This is a population isolate in South Africa who originally had come from the Netherlands. An independent study found the same SNP associated with increased risk of syncope or cardiac arrest. And a study led by Yalda Jamshidi of 58 cases of torsade compared to controls showed that SNPs highly correlated with the ones I just showed you are also associated with increased odds of torsade in response to drug exposure. So hopefully by showing you these two examples, SNPs in the back of the room, but I'll tell you the effects per allele are about 3 milliseconds. There's one polymorphism that had about an 8 millisecond effect. We found common polymorphisms in the same long QT genes. This is not atypical. That common polymorphisms with weak effect on a continuous measure will also occur in the same genes that have rare mutations of strong effect that are associated with Mendelian disease. We expanded that genome-wide association study to reach about 70,000 people. This is another large study, 230 co-authors. I can't thank everybody, but Don Arking at Johns Hopkins, Sarah Pewlett was a computational student in my lab, Paul de Bakker at the Broad, now in the Netherlands, and Arnep Pfeiffer at Technical University of Munich. Just a few shout-outs of people who were critical to the study. This shows the association results across the genome. It's 2.5 million results, where what we've done is placed each single SNP, it's a dot on this line, on the line of what you would expect to be the p-value distribution under the null, where there's nothing biologic going on. You would expect half of all results to have a p-value of 0.5 or less. This is actually the negative log 10 of the p-value. So 1.25 million results are shrunk into this little segment of the line, and so on and so on. You would expect 0.1% to have a p-value of 0.001 or less. In fact, we observed many more results. These are the observed distribution of p-values. Many more results than we would have expected due to chance. This tells us there's something going on. It's not just the null. Whoa. Yes, I agree. We encountered a problem. So we observed an excess of results more significant than we would expect. If we removed all 35 of the genome-wide significant results we found in the study, there's still an excess of lower p-values than we would expect. This suggests that there's still some more stuff to be found. But we found in this study 35 loci, of which 22 were novel. There are 68 independent SNPs at these loci. Some loci have multiple SNPs that are independent of each other. They explain about 8 to 10% of the variance in QT interval. Genetics in general explains about 30 to 40%. So these explain about a quarter of, it's mostly a function of left ventricular tissue. So in collaboration with Mike Morley, Tom Coppola, Ken Margulies at Penn, they have a magnet consortium that includes heart samples that were biopsied at the time of either transplant, so very diseased heart, someone who needed a new one, or a potential donor whose heart was not used for whatever reason, but who was brain dead and died of a different non-cardiac cause. In those roughly 300 samples, either with heart failure or apparently healthy other than being brain dead, we examined all transcripts within a megabase of each of 63 SNPs that were present in their data. I said there were 68 that are independent. We used a multiple testing threshold that was 0.05 divided by the number of tests that we did. And in order to see if the association with expression was likely the explanation for the association with QT interval, we adjusted for the best expression SNP to see if our GWAS SNP association disappeared. You would expect if they were concordant that they would. And in fact, all of the SNPs listed here in bold are SNPs that had significant expression in the left ventricular tissue. This is the transcript association of the QT interval SNP after adjustment for the best expression SNP. And most of these go from p-values that are less than 10 to the minus 4 to p-values that are much greater than 0.05, consistent with the transcript association likely explaining the QT interval association. This prioritizes a set of genes, the genes encoded here for further work. It certainly could be that there may be other genes at the locus, but finding an expression effect certainly in the relevant tissue increases the likelihood of that gene being associated. We looked in data from the epigenomics roadmap and found that our 68 SNPs, or very close proxies, overlapped left ventricular enhancers in humans. This is work led by graduate students in Chen Wang, Manolis Kellis, and Laurie Boyer at MIT. And we found, compared to the null expectation of the number of times we would get a SNP that overlaps an LV enhancer, we observed it occurring about 37 times. This is decidedly outside the range of this curve, and it's a very significant p-value for enrichment of LV enhancers at these sites. We also screened roughly 300 individuals with long QT syndrome who did not have mutations in the top 3 genes in collaboration with a set of investigators, including Mike Ackerman, Peter Schwartz, Connie Bazina, Artur Vilda. Sequenced exons for 6 of our favorite genes at these novel loci, totally arbitrary, mostly driven by the fact that these genes are known to be expressed in the heart and or to underlie factors involved in myocardial physiology. And we compared those to controls as well as to exome sequencing project. I'll highlight 2 of the findings where we found premature stop codons in ATP2A2 that encodes SERCA2A. That's a regulator of calcium release from the sarcoplasmic reticulum. And TRPM7 is a recently identified cardiac calcium channel. It also has kinase activity. I won't go into detail about these experiments in the interest of time. This is just to remind you about the cardiomyocyte action potential. The action potential is underlaid by multiple different ion channel currents that are activated at different times in the action potential. Most of the upstroke here is due to sodium influx through a cardiac sodium channel. This plateau phase is due to balanced calcium influx. That's what causes contraction of the muscle. And then potassium efflux, which causes a repolarization to a negative potential. In fact, we were really surprised to find that so many calcium regulating genes occurred at our QT interval loci. So just to tick them off, the ATP2A2, that's SERCA2A, is a QT GWAS gene. Phospholamban is a negative regulator of ATP2A2 QT gene. Protein kinase C is a regulator of phospholamban, also a QT interval gene. ATP1B1 is the sodium potassium ATPase, also a QT interval gene. SLC8A1, the sodium calcium exchanger, also a QT gene. Sarcoluminin is known to be involved in calcium cycling. TRPM7, not really clear how it influences calcium cycling, but I would say this is a new observation that myocardial repolarization, there was one Mendelian calcium channel gene, but it does appear that the excitation contraction apparatus that influences calcium cycling and contraction is also related to myocardial repolarization. Whether this relates to heart failure having an increased risk of arrhythmias is not clear. And we are currently doing a genotype-directed physiology study under the hypothesis that if long QT syndrome mutations are strong risk factors for torsade, and 2 SNPs with relatively large QT effects increase risk for torsade, then maybe common variant genotypes in aggregate that prolonged QT interval might lead to exaggerated QT response to meds. And so we are currently, I'm not going to show this in the interest of time. I'll just say we have a QT score where if you take just the top 14 SNPs and you put them together in a score, it explains, as I mentioned, 6 to 8% of variance in QT. There's a 15 millisecond difference between the top 20th percentile and the bottom 20th percentile. I told you that drugs get pulled from the market for less QT prolongation than that. So we think it's plausible that this degree of genetic difference, now if we build a better mousetrap with 68 SNPs, will identify people at increased risk of drug-induced arrhythmias. And so we're recruiting healthy volunteers, selecting them based on their genotype score, giving them moxifloxacin, which prolongs the QT interval by about 10 milliseconds. We're about a quarter of the way through this. We are also two slides appeared twice. Giving people who are getting clinically determined dofetilide. Dofetilide is an antiarrhythmic that prolongs the QT interval and is associated with torsade. It has to be started in the hospital to monitor the EKG, and we're drawing blood from everybody who comes into any of 5 Boston area hospitals, and then genotyping them to see if these SNPs in aggregate influence the QT response. So hopefully I've shown you that rare genetics has been critically important to informing identification of drug targets and understanding drug toxicity. I think common variants will also play their role. Thank you very much. Happy to answer any questions.
So, can you hear me? Thank you, Chris. That was a wonderful talk, and I just want to sort of emphasize to the audience how amazing it is what you've been able to do. You bridge across the entire spectrum from basic to clinical translation. It's really impressive. You have 250 co-authors, but you're leading these studies. You're doing the epi, you're doing the clinical studies, you're doing the basic science. It's very impressive. We have time for a few questions, if anybody has any questions. Yes. Actually, the speakers up here will capture your voice for the screen.
All right, great. Yeah, a great talk. I was curious, at the beginning of the hypertension talk, you mentioned that 40 to 60% of hypertension was, driven by genetics. Yet when you go looking with the SNP approach, it's about 1%. Where do you think that missing, 40 to 50% is lying? Where's that at?
I don't know. I think the power estimates that we've basically had between 5 and 10% power to detect each of the common variant effects that we've found so far. So that would suggest that there are 10 to 20 variants that we haven't detected for each variant that we have found. So I would say a reasonable estimate of the number of common variants with comparable effect sizes is maybe 400 common variants. Probably there are thousands that have really minuscule effects. I think what the contribution of rare variants with very strong effects that are weaker than would make you be in a Mendelian family, but strong enough to have a pretty dramatic impact on blood pressure, we'll only know when we've sequenced enough people. We're probably 5 years away from that.
What about methylation, imprinting, that kind of thing? Something you wouldn't pick up with a sequencing approach.
Yeah, I think imprinting clearly could influence the heritability. I think these are common SNP arrays. There are gaps in what we can study with SNP arrays. Methylation experiments are ongoing. It's I think there's a lot to do to explain heritability. What I can say so far is common variants don't do a very good job as far as they've been discovered so far. But I can't really speak to what the future will tell us about what explains that variation. These heritability estimates are potentially confounded by shared environmental factors that can't be adjusted for. It's possible that as much as half of it has to do with how much salt you have in the diet, whether your family exercises, and whether your family smokes or sits around watching TV. There's a variety of factors that are not genetic but are transmissible.
So on that note, a lot of the work that we do in this omics arena, we're trying to sort of detect signal to noise, right? So I think your blood pressure phenotype, there's a lot of noise there.
Totally,
so
yeah.
How much, sort of where are you guys going in terms of improving the signal? So extreme phenotypes that aren't Mendelian or mapping response to a certain stressor, like where are you guys going with the phenotype? Because it seems like that's a big issue.
Yeah, I think it is. Well, so I'll say we'll continue to do these super large GWASs, and we'll find smaller and smaller things using our crude approach. I'm not stupid. These are not standardized. The ideal experiment would be you take a birth cohort of everybody who, let's say, is 40 years old. You put them on a standard salt diet for 2 or 3 weeks. You put an ambulatory blood pressure monitor on them. You get ABPM 24-hour blood pressure measures for, a day a week for 3 or 4 weeks. Then you take the average, right? That would be a much cleaner, more precise estimate. You'd wean people off their antihypertensives before doing that. We're not going to do that.
Yeah.
That is, you could estimate how much you'd improve the power of your study based on how much more precise blood pressure phenotype you get. You're still going to be in the probably 5,000 or 10,000 person study, and to do the kind of study I just described in so many people is prohibitive. So I think it's definitely one important lesson is with dirty phenotypes, if you can get enough people and you're not susceptible to confounding, and you have to deal with ancestry in these studies, you can find things that you otherwise just could never find due to the noise. If you picked an incredibly refined niche phenotype that only you have, because you're the only one who knows how to measure it, or it's got a lot of standardization, you're at risk of finding things that are maybe real, maybe not, and not being able to replicate. And so I would be very careful about your exit strategy from designing studies that are too refined. But I'm not going to, most of the hypertension community, thinks these studies are ridiculous. You find blood pressure variants. At least we're finding a bunch of known things that influence blood pressure. But I would readily acknowledge these are very dirty phenotypes. It requires, if you have a way to refine the phenotype or look at an intermediate, consider the plasma natriuretic peptide story, then I think you could study 50 to 100 people and have reasonable power to discern an intermediate effect.
I also want to sort of highlight to the group the to go from these big population studies, which you guys do at the DCRI from the outcome standpoint, from the clinical trial standpoint, and then really sort of going into these small-scale human physiology studies. And I think the genotype-guided approach is really interesting, and the work that you did was very elegant. I think that sort of, I just want to highlight to the group that this is really a novel cutting-edge approach to really understanding the physiology in a very human context. My question for you is, I can't remember if your SNP was coding or not, like when do you decide you've identified the right signal before you go to all the time and effort that it takes to do a genotype-guided callback study?
Well, we're doing a callback study. Well, so for the QT part, I said we had 68 SNPs. Maybe for that D85N amino acid altering polymorphism, that's probably the SNP.
Right.
Probably not another one. But for a lot of those other non-coding ones, it might be causal. It might be a SNP that's 10 kilobases away that's highly correlated, but it might not be the same one. If it identifies a group of people who have a differential QT at baseline and a differential predisposition, I don't really care.
Okay.
I'm not trying to find the causal variant. I am trying to find the causal variant for some of those loci. But things that will make me more enthusiastic about pursuing molecular experiments would be this very strong expression effect. Protein kinase C is an example where the expression effect is like 10 to the minus 20 in 300 samples. It's probably acting through that. It seems reasonable to go after that gene as a potential target. For the blood pressure scenario, we're following up on the soluble guanylate cyclase gene. There's one SNP that has 10 correlated SNPs in the interval, and we're going through SNP by SNP and building luciferase reporter cardiac enhancer assays for each of the 2 alleles for each of those SNPs, including a kilobase around them. So that's like 20 different constructs to build and test. So it's laborious. I think we're doing it for the SGC locus because SGC seems important. It's a drug target. It seems it's a targetable pathway. It seems like there could be a lot of early clinical traction if we could really nail down the mechanism by which it influences hypertension. Then maybe people would be more motivated to go after it as an antihypertensive target. So it's very specific, but I would say most of the stuff that we publish, we may have a little flag with a little gene label because that's the gene next to it. Right. But until you get some good evidence that that's the gene that influences it, you should be very cautious.
Okay, thank you very much.
Great,
for
thanks very much.